Our laboratory is working on a unique research concept to understand the molecular mechanisms of synaptic transmission. Our research is based on a cycle of solving important unsolved problems in neuroscience by developing technologies to tackle our questions, and then developing further technologies to tackle the new problems we discover. We expect this approach solves important problems that no one has been able to solve before and brings us closer to the truth. Our lab is working on the following research topics. First, we are developing new fluorescence imaging techniques. We are developing high-performance fluorescent neurotransmitter probes as tools to observe the spatial-temporal dynamics of neurotransmitters in living cells, which no one has been able to observe before. We are also developing next-generation fluorescence labeling techniques to specifically label target molecules in living cells. With these techniques, we have made findings by actively introducing chemical biology methods to neuroscience. Second is the study of synaptic transmission using our unique fluorescence imaging methods. Here, we are working on neurotransmitter imaging and visualization of the nanoscale arrangement of synaptic molecules by super-resolution microscopy. By integrating these techniques, we are trying to solve important problems in synaptic transmission. Let us introduce hybrid-type fluorescent probes as an example of our unique fluorescence imaging techniques. We are developing fluorescent probes that have both high specificity and sensitivity to target molecules by labeling a small organic fluorophore to a protein that specifically binds to the target molecule. Here, we incorporate chemical biology methods to rationally design and create the probes. Hybrid-type fluorescent probes drastically change their fluorescence property by binding to target molecules. We have developed hybrid-type probes for ADP, glucose, and glutamate and are applying them to neuroscience research. Here's an example of the application of our technology to synaptic studies. By visualizing glutamate released from central nervous synapses, we can understand how these neurotransmitters are released from presynaptic terminals. We also use super-resolution microscopy to understand the molecular mechanisms of synaptic function, focusing on the nanoassembly of synaptic molecules. We have constructed a custom-made super-resolution microscopy system and have successfully observed the nanoassembly of synaptic molecules with the world's highest spatial resolution. By using these technologies, we have revealed for the first time that the nanoassembly of synaptic molecules regulates the efficacy of neurotransmitter release at the presynaptic terminal. We will continue our research to elucidate the relationship between the spatial temporal dynamics of nanoassembly and higher brain functions. Why don't you join us to do unique neuroscience research using your original technology? Please have a look at our website and feel free to contact us.